Mature B cells can also be rendered self-tolerant. It is likely that most self-reactive B cells will encounter their antigens while still immature, as many self-antigens circulate through tissues in soluble form or are expressed by many different cell types, including those in the bone marrow. Nonetheless, some self-antigens are not present in the tissues through which immature B cells pass, and thus B cells expressing receptors specific for these antigens will survive to become mature. Lack of T cell help or an inability of the antigen to strongly cross-link the B cell receptor will usually prevent these cells from becoming activated and causing problems. However, mechanisms for eliminating or inactivating mature self-reactive B cells and even activated self-reactive B cells also exist. For example, B cells expressing immunoglobulins specific for H2KB, MHC class I molecules are deleted even when, in transgenic animals, the expression of the H2KB molecule is restricted to the liver by use of a liver-specific gene promoter. B cells that encounter strongly cross-linking antigens in the periphery undergo apoptosis directly, unlike their counterparts in the bone marrow, which attempt further receptor rearrangements instead. The different outcomes may be due to the fact that the B cells in the periphery are more mature and can no longer rearrange their light chain loci. On the other hand, stromal signals may be important. Recent data suggests that stromal cells in the spleen instruct B cells to respond in a different way to signals delivered through the antigen receptor compared with the stromal cells of the bone marrow. Mature B cells that encounter and bind an abundant soluble antigen become energized. This was demonstrated in mice by placing the HEL transgene under the control of an inducible promoter that can be regulated by changes in the diet. It is thus possible to induce the production of lysozyme at any time and thereby study its effect on HEL-specific B cells at different stages of maturation. These experiments have shown that both mature and immature B cells are inactivated when they are chronically exposed to soluble antigen. Finally, there may be a mechanism to eliminate activated B cells if they mutate to self-reactivity while they are proliferating and undergoing somatic hypermutation in germinal centers and peripheral lymphoid tissues. This possibility has been demonstrated by first stimulating a germinal center response by immunizing with foreign antigen, and then infusing large quantities of this antigen in soluble form, a procedure that induced apoptosis in the responding B cells. This experiment was designed to mimic the situation in which a germinal center B cell alters its specificity to express a mutated immunoglobulin with high affinity for self-antigen. However, whether this form of self-tolerance operates under normal conditions is not yet clear. And that's mature B cells can also be rendered self-tolerant. Have a great day.